LAC OPERON. LAC OPERON. This is E. coli. This is E. coli. E. coli. E. coli is having LAC OPERON. Okay. E. coli utilize glucose, but in adverse condition it can also utilize lactose. I say E. coli utilize glucose, but in adverse condition it can also utilize lactose. For utilization of lactose, lactose is disaccharide made up of glucose plus galactose joined by beta linkase, glucose and galactose joined by beta linkase, and the name of enzyme is beta galactosidase. So lactose can be utilized by E. coli, but E. coli require an enzyme beta galactosidase. Beta galactosidase is an enzyme. All enzymes are protein, and you all know DNA is there, DNA to RNA, RNA to protein. Yes, I have already told you that DNA can't do work. It is Malik. It synthesize messenger RNA do synthesize protein do work. So if beta galactosidase is there, there must be DNA for beta galactosidase, there must be gene for beta galactosidase. We saw that in E. coli there is a gene, that is LAC-Z gene. There is LAC-Z gene, that is having this gene. When synthesize messenger RNA, synthesize a protein, this protein has beta galactosidase activity. So I can say, again I repeat, E. coli can also utilize lactose, but lactose is a disaccharide. If you want to utilize lactose, you must have beta galactosidase activity, because glucose and galactose are joined by beta linkase. Beta galactosidase activity is present in a protein. This protein can be synthesized from a gene. If this gene is present in E. coli, that gene synthesize messenger RNA protein. Yes, this gene is LAC-Z gene present in E. coli. When synthesize messenger RNA, it synthesize a protein, that is beta galactosidase. We found that if E. coli want to utilize lactose, it must contain one more protein, that is permease. One more protein, that is permease. Okay, this permease, what is the function of this permease? This permease protein attached to the membrane of E. coli, and with the help of this permease, lactose enter the E. coli. With the help of permease, lactose enter the E. coli. Normally lactose can't enter E. coli in large amount, but with the help of permease it can cross E. coli cell membrane and can enter and can be utilized. Clear? So other than permease, E. coli also require one enzyme, transacetylase. So there are three enzymes in E. coli, or there are three genes, ZYA, Joya, ZYA, mnemonic बना दिया हमने, कुछ भी, you just remember ZYA. Z is responsible for the synthesis of messenger RNA, which will synthesize beta galactosidase. Y is responsible for the messenger RNA, which will synthesize permease. A is responsible for synthesis of RNA, which will synthesize transacetylase. With the help of these enzymes, E. coli can utilize lactose. So we found that in normal situation, whenever there is sufficient amount of glucose, you all will give me answer, because I will come to know that how class is going on. Okay, or is it monotonous, or you are enjoying, or what is the status? I know that genetics little bit difficult, and if we add little bit more concept, it becomes little bit boring. Okay, yes, question is, if glucose, yes, in if glucose is sufficient, is there any need to utilize lactose? Please do reply. If there is glucose is sufficient, is there any need to utilize lactose? Yes, there is no need to utilize lactose. Yes, there is no need to utilize lactose. So at that time, these genes must be kept in inhibited state. Yes or no? Yes. Means when there is sufficient amount of glucose at that time, E. coli don't want to utilize lactose. To utilize lactose, E. coli must have these three enzymes, but when there is sufficient amount of glucose, these genes must be kept in inhibited state. So what happened? We found that near to this gene, yes, near this gene, yes, there is operator region. Okay, function of transacetylase, in few places it is written is not well defined, but at few places it is written that when lactose will enter via permease, some toxic compound will also come in E. coli. This transacetylase will destroy these toxic stimuli. Although it's not asked, question is not asked, but you asked me, so I told you that in few books you will find standard textbook that transacetylase is not having well defined function, but few textbook says that the function is to detoxify the compound which will come inside the E. coli along with lactose with via permease. Okay, dear? Yes. So we found that how these genes are regulated. We found that in E. coli there is a gene that is LAC-I. This is LAC-I gene. I means inhibitor. I means inhibitor. This LAC-I gene, I for inhibitor, what it do? It synthesize messenger RNA, synthesize protein. These proteins are repressor protein. These proteins are repressor protein. These proteins are repressor protein. Four repressor protein join together to form repressor tetramer. We say LAC-I gene is constitutional gene. Or housekeeping gene. Yes, I can understand, dear friend. Yes, what you will do, I can understand that these topics are not studied by us during our preparation time and will remain untouched. Because in biochemistry, whenever students they start with carbohydrate, lipid and protein, they become tired and they leave. Okay, till we reach genetics, that it becomes difficult. So what you will do, if you are getting some problem, when you will listen it once in increase speed, then you will find that these concepts are really useful. Okay, so I say that LAC-I genes are constitutional gene or housekeeping gene. You must have seen in hotels, housekeepers, they come again and again and do clean the room and everything is adjusted. So this is housekeeping gene, means they are continuously doing work. They are doing work continuously. So these are constitutional gene, housekeeping gene. Yes, means they are expressed every time. Just like in human being, if I say hexokinase, hexokinase is required for glycolysis, and this hexokinase is present in each and every cell of our body. So hexokinase is constitutional gene. Hexokinase is an enzyme. Enzyme are synthesized from gene. So gene for hexokinase is constitutional gene. But glucokinase in liver, pancreas, when we blood glucose is increased, then we require, and that's why they are inducible genes. So yes, these are housekeeping gene and inducible gene, two types of. Okay, so this is constitutional gene. Every time it synthesizes repressor protein, these repressor proteins synthesize tetramer. This tetramer goes and bind to operator region. Tetramer goes and bind to operator region. Okay, repressor tetramer goes and bind to operator region. Okay, this RNA polymerase, this is operator. How you will remember? O for operator. O looks like, sorry, repressor tetramer, repressor protein looks like O. So these repressor tetramer bind to which region? Operator region. Okay. Yes, repressor tetramer bind to operator region. RNA polymerase bind to promoter region. RNA polymerase bind to promoter region. Okay. Yes, constitutional or and housekeeping gene are same. Yes, constitutional gene or housekeeping gene. Sorry for that. Or housekeeping gene. Okay. Constitutional gene or housekeeping gene are same. This repressor tetramer go and bind to which region? Operator region, which bind to operator region. RNA polymerase is attached to P4 polymerase, P4 promoter region. I have told you earlier. Yes, RNA polymerase with the help of sigma 7 it bind to promoter region of DNA. So it bind to promoter region of DNA. RNA polymerase is attached to promoter region of DNA. This enzyme is RNA polymerase. This enzyme is RNA polymerase, and this RNA polymerase can synthesize RNA, but this operator is attached with the repressor tetramer. Yes, now till now you know everything little bit about E. coli genome LAC operon. Yes, we found that E. coli normally utilize glucose, but in adverse condition it can also utilize lactose. So we found that to utilize lactose, all the genes which are required for the utilization of lactose are present in E. coli in a linear sequence. All genes for utilization of lactose are present in a linear sequence. This is called LAC operon. Operon means operating unit. This operon model was given by Jacob and Monod. Clear? Now this we found that this LAC operon operating unit, yes, have LAC I gene. This site I have not discussed till now. This site is called cap site. Why it is called cap site? This portion of DNA is called cap site. Why this is called cap site, I will discuss shortly. For the time being, Normally this is lac gene. Every time when this is being expressed, synthesize repressor tetramer go and bind to operator region. RNA polymerase bind to promoter region. RNA polymerase want to synthesize this messenger RNA. RNA polymerase want to synthesize this messenger RNA but can't synthesize. Why can't synthesize? Yes, because this operator region is attached with repressor tetramer. I told you it slides over it. Yes, you must have seen that how this it slide. But if repressor tetramer is there, if repressor tetramer is there, then if repressor tetramer is there, it will not be able to synthesize. It will inhibit it. Okay, clear? So normally this RNA polymerase will not synthesize this messenger RNA, and actually this is not required. Because glucose is present. Now what happened? Glucose decreases in environment. When glucose decreases in environment, cyclic AMP increases in E. coli. Please do reply. In our body, if glucose decreases, cyclic AMP increases or decreases? Please do reply. In our body, yes, in our body, when glucose decreases, cyclic AMP increases or decreases, and how it is regulated? Definitely, we developed from E. coli, bacteria, gradually crores of years. So, same concept. Glucagon, same concept. Glucagon increase cyclic AMP in us. So E. coli will follow the same rule. When glucose decreases, cyclic AMP increases. Cyclic AMP will bind with CAP. CAP is catabolic activator protein. CAP cyclic AMP complex. This CAP cyclic AMP complex will go and bind to CAP site. So now what happened? Glucose decreases in environment, cyclic AMP increases in E. coli, CAP cyclic AMP complex is formed, it goes and bind to CAP site. This CAP site activate RNA polymerase. This CAP cyclic AMP complex activate RNA polymerase. Repressor tetramer don't allow. Now, suppose you are RNA polymerase. I ask you, you are RNA polymerase and repressor tetramer is your best friend. Yes, it is living with you for such a long time. Yes, here if I forget about this scenario, I told you that RNA polymerase is attached with promoter region, repressor tetramer is attached with operator region, you are living together, you are best friend. Yes, he said no, don't synthesize messenger RNA. Now CAP cyclic AMP came, girlfriend came. CAP cyclic AMP said please do synthesize messenger RNA. Then whom you will follow? Your girlfriend, CAP cyclic AMP, or your best friend, repressor tetramer? Yes, whom you will follow? You will follow your girlfriend. Definitely, I have seen many times. Yes, you have seen so many times in your life that so many close friendships are broken by girlfriends. Okay, yes, it's sometimes girlfriends are good. That yes, this cyclic AMP indicate glucose is not there. Oh, Ravi. Yes. This is the real friend, huh? You should. Yes. Yes. This RNA polymerase will follow cyclic AMP, because if cyclic AMP is increased, it means glucose is not there. Either synthesize or E. coli will die. Either synthesize beta galactosidase or E. coli will die. So this RNA polymerase will follow cap cyclic AMP complex. But your friend, yes, your friend is very चिपकू फ्रेंड, यू नो. यस, ही सेड नो नो नो, तुझे मेरी लास्ट पे से जाना होगा. You can't go, you can't go, I will not let you go. Yes, he is standing here, I will not let you go. Now what he will do? He said तुझे मेरी लाश पे से जाना होगा. उसने कहा ठीक है, मैं लाश पे चला जाऊंगा. He jumped from the lash of this pressure tetramer. He jumped and synthesized. But he would be sad. But you would be sad. Yes, you jumped, you left your friend, but you are sad because but you have to synthesize, otherwise E. coli will die. So you will synthesize messenger RNA. You will synthesize messenger RNA. This messenger RNA will be synthesized at very slow speed. What I said? Yes, synthesize at very slow speed. But when this messenger RNA would be formed, beta galactosidase will be formed, permease will be formed. Yes, initially beta galactosidase will be synthesized in very little amount. Permease will be synthesized. Permease will bind to the cell membrane. Lactose will enter. Lactose will enter. This lactose will act as inducer. What I said, lactose or allolactose will act as inducer. Inducer will bind to operator region and will take it back. Yes, will bind with inducer. Lactose will bind with, sorry, lactose is acting as inducer, bind with repressor tetramer and separate it from operator. And when it will separate it from operator, synthesis will rapidly increase. So a question was asked. Have you understood this part? Yes, a question was asked. They gave question like this, that in a solution, yes, this y-axis is beta galactosidase activity. Yes, this is time. So this is beta galactosidase activity. Suppose beta galactosidase activity is there. At this point, glucose decreases, cyclic AMP increases. Cyclic AMP increases. This cyclic AMP increases. As cyclic AMP increases at this point, after that, yes, there would be little increase in the beta galactosidase activity, and after some time there would be great enzymatic activity, beta galactosidase. So cyclic AMP increases at this time, but to achieve maximum beta galactosidase activity, yes, to achieve maximum beta galactosidase activity, there would be a lag. Why this would be a lag? Initially beta galactosidase will be formed, permease will be formed. When permease will allow lactose will act as inducer. It will separate repressor tetramer from here. Then RNA polymerase will do rapid synthesis and beta galactosidase activity will increase rapidly. Please do reply. Is it clear to you that at this point, yes, cyclic AMP will increase? And if I ask you what happened at this time, please do reply. What happened at this time? Yes, at this time, removal of repressor tetramer. Repressor tetramer. Removal of repressor tetramer. Yes, very good. Lactose bound to tetramer or removal of, yes, lactose enters. Permease increase and lactose enters. Yes, lactose enters into cell and separate repression tetramer, so that activity increases. Very good. Means you are understanding the basic concept. Yes. This question, this is very difficult question. Recently asked in MLE. Okay. And they ask question and that can be repeated. In LAC OPERON is not untouched part of our exam. Okay, so you must know. So you can know the concept. LAC OPERON operating unit. OPERON model was given by Jacob Monod. Now you remember girlfriend is more important than boyfriend. Yes, you have seen so many movies. So yes, cyclic AMP is more important than repressor tetramer. Yes, cyclic AMP is more important than repressor tetramer. This idea of boyfriend girlfriend just came to my mind this time only when I am teaching you. Okay, and next time you will find animation on this also. Yes, लाश भी मिलेगी तुम्हें friend की. You will get the dead body of friend and he will jump from the dead body. Girlfriend would come. Okay, then yes, now this would be an interesting story. Okay, yes. So hope you have understood the LAC OPERON and you will be able to solve many questions by this cyclic AMP. Cap cyclic AMP or cap cyclic AMP is more important than repressor tetramer. If both are there, you will follow cap or repressor tetramer. Yes, you will follow cap or